Okay, our next speaker is Dr. Wai Wong, who I'm delighted to introduce here. He spent many years working at the National Eye Institute, and then more recently has shifted to work on preclinical drug development. And today he'll be speaking about targeting microglia and neuroinflammation in retinal diseases.
Today I'll speak a little bit about targeting microglia and neuroinflammation in retinal disease. I was glad to know that there are ongoing programs looking at the same thing here at Penn, so please hold me to account if I get anything wrong. So today I'll focus a little bit as to why we want to target microglia and neuroinflammation at all, and what is the evidence that dysregulated neuroinflammation and aberrant innate activation can contribute, if not to the cause, then to the progression of retinal pathology. I'll give a very brief primer as to the cells that comprise the innate immune system that live within the retina or come into the retina during disease. And also to survey broadly the targeting strategies and mechanisms that are out there in potential therapies, classifying approaches from the broad to the narrow approach, going to something that extends across all innate immune cells to those that go after just one specific mechanism. And again, finally, at the end, review the competitive landscape for current approaches in the clinic right now. So just a very brief primer, perhaps not needed in this case, where microglia in the retina are the prominent part of the resident glia population. And you can see them here in green and on the right as ramified cells that live in the IPL and the OPL and the branch across the entire horizontal aspect of the retina, covering all retina areas. Unlike any other cell within the retina, these processes of microglia are dynamic under normal conditions, and they constantly survey extracellular space, coming into close contact with retinal synapses and vasculature, and indeed, as we found out, play roles in synapse maintenance and also vascular maintenance under normal conditions. However, under conditions of disease and injury, these cells don't remain where they are. They become very activated, change in morphology and the physiology, proliferate, and then migrate the sites of injury in the attempt to restore homeostasis. But as we'll find out, they actually can have maladaptive adaptations also. So these are the cell types that make up the overall innate immune cell population. They include microglia, but also in the conditions of disease, we see monocytes from systemic circulation moving into the retina and taking up residence as macrophages. And just beyond the blood retinal barrier in the choroid, beyond the RPE cells, there's also a rich population of choroidal macrophages that are in between the the choroidal vasculature, and also play a role in maintaining immune function within the outer aspect, just beyond the RPE cells. And all these three cell types do come together in pathological situations to shape the overall progression of disease. The last cell type here is the Mueller cell. They're not part of the innate immune system. However, they do, as our work in the lab has shown, modulate interactions between Mueller cells and microglia cells and macrophages to shape the overall dynamics of immune responses. So these are the players that come to the fore in the conditions of disease. And why do we think that going after microglia might be a good thing to do? What's the implication of microglia in retinal diseases? This is a busy slide, but I'll just summarize here. Across diseases like glaucoma, age-related macular degeneration, and also diabetic retinopathy, we do find microglia at the scene of the crime. At the site of pathology, we see the accumulation of cells there, and certainly they're present in human histopathology in different stages of disease. Secondly, we also see the implication of genes that are expressed by microglia cells in GWAS studies, particularly in AMD genetics, where inflammatory and specifically complement genes are highly implicated. And these genes are expressed by microglia cells in large proportion. Number three, there's also a preclinical validation in models of retinal disease of all three kinds, and there's some in the poster session out there, where interventions that target microglia cells can change and ameliorate the outcomes of diseases in these animal models. And last of all, clinical validation. We know already that modulating the innate immune system through steroids has been effective in diabetic retinopathy. And also more recently, the modulation of complement has been also been approved in the form of drugs for geographic atrophy. So this conglomeration of data does suggest that going further in this direction might be fruitful for the conditions that still remain untreated. To summarize how we can actually go after microglia activation and neuroinflammation, there are many different approaches that have been taken. Sorry, I didn't parse too well. But I've arranged this in the overall pyramid from the very broad approaches, where the entire innate immune system is being modulated, to very specific approaches, narrow approaches, where a single effector mechanism is intervening that without changing the innate immune system themselves. And taking it from the very top, the broadest way that you can go after microglia is actually to deplete the entire population. Indeed, microglia cells are very sensitive to signals that are required for their survival. If you block CSF1 R signaling, you can get rid of all the microglia cells in the retina in a very short order. And this may be, as our lab has found in last years, that this may not be a great strategy, because although the microglia are not required in the short term for retinal function and structure, if you sustain depletion over weeks to months, we do find that the retina sustains synaptic degeneration, loss of ERG function, as well as in terms of choroidal atrophy and thinning. There's also a decimation of the overall thickness of the choroid, as well as the vasculature in the choroid capillaris. So there is a requirement for innate immune function for homeostatic maintenance of synapses and vascular structure. So a very broad approach of sustained depletion may not be possible from a safety standpoint. However, there are lighter touches that you can imply, such as transient depletion. In some published data work from our lab a few years ago, we found that if you deplete microglia pharmacologically in the retina and stop the pharmacological depletion, these cells can spontaneously regenerate in a matter of days, as seen in the middle of the slide there. They can, again, repopulate the retina as if depletion had not occurred. This is true not only in the retina, but also in the choroid. As shown in the lower part of the slide, you can actually deplete all the choroidal macrophages, and they too can come back quite quickly in a matter of weeks. So this may be a way that you can sort of hit reset on the overall innate system within the eye to deplete all the maladaptively programmed microglia and enable repopulation, as it were, from fresh cells. There is also an approach where you can, as we have shown in a paper we published last year, where you can transplant iPSC-derived microglia into the retina, and they too will take up their on the typical positions within the retina. And so this may be a way to get rid of all the microglia and then introduce the kind of microglia that you want, out with the bad and in with the good, in order to reset the entire immune system. And then there's another approach that's been taken by Florence Senlob's lab in France, which is to get rid of not all the microglia, but a specific subset of microglia, the subretinal microglia in AMD. And he's formed a company to go after CD47, which is a receptor that can trigger the selective death of macrophages in the subretinal space, but not affect the other microglia within the retina through truncated TSP1 protein. So these are the approaches that have been broadly looked at. And with respect to more narrow approaches, you can keep the microglia where they are, but you can change the polarization through either changing their environment or pressing buttons on their cell surfaces to get them to move from one polarity to another. And in the slide on the right, you can see that microglia cells are between these two states, the homeostatic, which is the good microglia, and the maladaptive microglia, which may drive pathology. There are signals in the environment of the microglia that can impinge on the microglia, as well as these receptors that can shift them towards the green or towards the red. And this approach has been taken in a number of ongoing studies. There is ongoing GA study by Avicida, where nanoparticles introduced by IVT injection is able to trigger cyclic receptor activation to shift the state towards the M2 resolution state, as they have explained in their literature. There's also a set of assets that have been developed to target microglia along the ATP, PRX, P2RX7, NLRP-mediated sequence, where there are treatments that can reduce the expression and the secretion of ATP through connexin channels, and also inhibitors on the P2X7 channel to prevent activation through ATP. And of course, there's also a number of programs, most specifically that by Inflammasome Therapeutics, where the downstream intracellular activation cascade is interrupted through the NLRP3 inflammasome, currently in clinical trials right now. So these are some of the approaches that are currently being undertaken to try to modulate microglia from the inside by switching the overall polarization state. Down to the very tip of the pyramid, you can leave the microglia cells as they are, but simply interrupt the ability to signal through cytokine signaling, through complement signaling. And certainly these cytokines and complements have been implicated in a number of different programs. And certainly we see that in the clinical trials ongoing right now. This is just a very busy slide to say that complement specifically is one such target that's attracted a lot of drug development attention. There are various targets along the complement cascade that have been investigated. They have succeeded in resulting in approved drugs, such as those highlighted in green, the ones that have failed, those highlighted in red, and those that are still ongoing. You can see there's a lot of activity in this area. And all this to try to change the overall signaling of complement within the retina to change the outcome of the disease. And certainly we're looking at this very closely to be able to find a path to even better therapies by looking at different complement targets, different combinations of targets, as well as focusing on some upstream targets that are ahead of complement activation. There is also a lot of activity in trying to understand which are the routes of complement inactivation that are most successful, such as going after systemic versus local complement activation. And you can see on the right that there's a whole lot of different platforms that have been used to go after complement, including small molecules, large molecules, antisense oligos, as well as gene therapy. And I think that this field will be a site of a lot of further development in trying to identify the best platforms and also the best systems and best targets. To summarize, why I'll be interested in looking at neuroinflammation, it's because it represents a therapeutic opportunity. Neuroinflammation is a contributor to pathologic features that are prevalent in retinal diseases. It's been linked to retinal degeneration atrophy, as occurs in GA, abnormal vascular permeability and neovascularization, as found in DME and wet AMD, as well as fibrosis, which is the cause of permanent vision loss in wet AMD. There's been remarkable advances in understanding of molecular targets for underlying these neuroinflammatory mechanisms, so that these can be then targeted through various molecular approaches. I think that we'd like to build on the clinical validation that exists for the approved anti-complement drugs, as well as steroids for DME and wet AMD, but to do so in a much more selective as well as safe way for better treatments. So the path towards more efficacious, safer, and less burdensome treatments does require us to consider additional targets and combination of targets potentially. There are some targets out there to go after, both complement as well as IL-6 inhibition, for example. And also we need to understand what's the best way of modulating overall innate immune system. Is it through systemic approaches or locally derived approaches? And I think there's a lot of development also in different platforms that give us a lot of capability. We heard about ASLs, we heard about gene therapy today, and being able to apply that not only to IRDs but also to common retinal diseases is probably the wave of drug development for the upcoming future. So I'll stop here and thank you for your attention.
Questions?
Yeah. Yeah, I think that recognizing that there is all these features and recognizing that the ways in which you can use a very broad, heavy-handed approach versus very specific approaches. How do we choose across that spectrum? I think this probably has to depend on the nature of the disease specifically, as well as possibly even the stage of disease. So I think that that's another fancy way of saying I don't know the answer to this question. But I think that as we understand a little bit more about how how the cells come at different stages, we can then design that approach to be able to say this mechanism is actually responsible for a lot of the pathology that is generated. Let's go after that and leave the rest alone so that it can continue to function positively. Or in the context of an intervention like gene therapy in the subretinal space, you really want to eliminate the majority of microglia in that area, so that you get rid of this pathologic aberrant response to the therapy you're trying to preserve. So I think that this will probably depend on the context, and as far as understanding context and recognizing what kinds of different tools you have, then allow you to match the tool and the mechanism to the context. But recognizing that you have choices is probably the first step to go. Yeah.
Hi, wonderful talk. Always lovely to hear. When we think about the NLRP3 inflammasome, it's an obvious target. Do you think that it's sufficient to knock out the NLRP3 inflammasome, or do you think that many of the inflammatory signals come from different pathways?
I think the trial has started in phase 1. This goes after using NRTIs to non-canonically inhibit the NLRP3 inflammasome. I guess the experiment has started. We'll find out the answer to whether it does work. It is a challenging target. It's intracellular in nature, and I think that there is probably a lot of consideration for the fact that it is a major pathway for activation. So if you took that away, would you have removed most of the badness that this pathway is contributing to? And have you also preserved the homeostatic aspects of it? The amount of scientific data, is probably not enough to really comment on the potential success. There have, of course, been success in preclinical models, but whether it would be actually be successful in the disease like AMD at this stage that is GA, I think we'll find out quite soon from the ongoing clinical studies. Yeah.
Thank you.
We have time for one or two more questions. That was a beautiful talk. When most people who transplant cells are intentionally depleting the immune cells prior to transplantation, given the data that you showed about transplanting iPS-derived microglia and as a reset button, do you think we should be transplanting microglia with ganglion cells or photoreceptors?
I think that we're just beginning to understand what it takes to transplant microglia. I think one aspect that really caught us by surprise is that microglia have their homing mechanisms, so that when you deplete them, the new ones can actually be generated and go to the right places and recapture this situation so perfectly that you don't even know that depletion has taken place. The same is also true for introducing iPSC-derived human microglia. We have introduced human cells into a mouse, and they too are able to find a place and live alongside endogenous mice microglia. They look a little bit different, but they actually form the same mosaic. So I think that we can, we're in the early phases of understanding what the possibilities are, but it's advantageous that these cells have the ability to home and find their location and be quite long-lasting. So cell-fate mapping studies have shown that once you transplant human microglia in a mouse, they actually are there sustainably for up to 6 to 8 months. So I think that this is just the first clue that perhaps we can sort of do this. I think the next phase of development will be to somehow modify those microglia to be able to play some role, be it express a neurotrophic factor, be it being biased towards a homeostatic role through some mechanism. So I think that that's probably the first sort of experiments that have to be done. In terms of combining them together with other transplantations, I think that that's a great idea. And I think that we'll probably have to learn more about how the simple experiments will work, and then maybe pave the road towards the things that you think about.
Why it's great to see you back here after having done your residency here and become faculty at NIH and become one of the top experts in microglia and macrophages. So my question now is there anything in general that you can say about blood-borne monocytes that become macrophages in the retina? Are they usually helpful, usually harmful, or context dependent?
Yeah, I think that it's um, probably context dependent. I think that also we don't know a lot of solid answers regarding whether the ones on the inside are good, the ones that come from the outside are bad. But I think the majority of the data does suggest that when they come into the retina, they are also quite similar in many ways to each other and respond to the same treatments. I was just speaking to Ryan and Ben, and that was also reflected in the profiling that is coming out from those studies. So these recruitment of monocytes could be just an augmentation of the mechanisms that microglia are already putting into place. And so you get more of the same through recruitment from the outside. I think the duality of we need one, not the other, one's bad and the other's good, is probably not as supported right now, although that possibility in some context still may be possible. I think a lot of treatments are being thought of as do we need to go after them locally, just treat the eye, versus systemically, where we treat all the innate immune cells within the eye, within the body to like monocytes. I think that question is relevant to the question you just posed. But I think that we will probably have to find out a lot more about that. But my gut reaction is that monocytes is sort of more of the same rather than duality of good and bad.
Thanks so much.